Okay, so really happy and thankful and excited to have Pat Secor here today to speak to us. Originally, Pat was nominated to give a seminar in the um, graduate training grant seminar series here, and due to some scheduling conflicts and some availability, we couldn't get him out here in person, but we were still able to host him here in the Ecology and Evolution seminar series, which is really awesome. Um, and so just a little background. So Pat runs a lab at the University of Montana in Missoula um, in the Division of Biological Sciences. Before he opened his lab there, trained as a grad student at Montana State in Garth James's lab, and then went on and did a couple of postdocs at the University of Washington in Pradeep Singh and William Parks's lab. I would say broadly, they study phage, bacteria, host interactions and how those interactions shape the evolution of both the bacteria and phage, and how those interactions shape their pathogenicity. So won't blabble on for too long and let Pat tell us about their work in the lab. So thank you. We'll hand it over to you.
All right. Thanks, Killian. For the invitation and for being flexible with me. I really appreciate the opportunity to speak with you. And I am bummed that I couldn't be there in person, but you know, I'm glad to be here on Zoom. So there it goes. It's not advancing for me, just like we said. I'll just click here. All right. So one of the pathogens that my lab studies is Pseudomonas aeruginosa, and it's an opportunistic bacterial pathogen. And it's pretty much everywhere in the environment. And as long as you're a healthy individual, it has a really hard time causing disease. But if you have some sort of underlying condition, it's really good at causing infections. And so some examples are chronic wound infections, diabetic ulcers, pressure sores, stuff like that frequently get infected with Pseudomonas. Another major group of patients that gets infected with Pseudomonas are cystic fibrosis patients. So Pseudomonas is a major airway pathogen in that patient population. Pseudomonas is also good at causing hardware infections or contact lens infections. Pretty much any opportunity it can and will cause an infection. Um, so how this relates to phages is if you look at the genomes of Pseudomonas strains, the majority of them are already infected by this PF phage. It's a filamentous prophage, and so it's already integrated into the chromosome of the bacteria. And when it is induced, it pops out of the chromosome, kind of replicates like a plasmid. And then from this plasmid, it makes a bunch of phage proteins and makes these filamentous virions that are extruded from the cell. And then it can complete its life cycle by attaching to type 4 pili, which are really structurally similar to the virion. And then as the pili are retracted into the cell, the virus follows, and then it can inject its DNA and complete its life cycle. And down here on the bottom is an EM image of a filamentous phage. In our laboratory strain PA01, it has a PF phage called PF4, and it's integrated right there. It integrates into a tRNA gene, which is a common insertion site for lots of viruses. And if you take the genome sequence of these PF phages that infect diverse Pseudomonas isolates and compare them to each other, they separate out into two lineages. And the thing that separates them out mostly is where they integrate into the chromosome, which tRNA gene they integrate into, plus a few other things. But the main point here is that these things are everywhere. They're a really common part of the Pseudomonas genome. So most viruses like to package their genomes into the most dense configuration, right? But that's not the case with these filamentous phages. They do the opposite. They package it in the most extended conformation possible. And so these phages, they are composed of thousands of these negatively charged coproteins that encapsulate a single-stranded circular DNA genome. And instead of mashing it into a small as small a volume as possible, you end up with these super long virions. So the genome is really small with these phages, like 10 kb, but the virion length can be up to 2 microns long, which is the same length as the bacterial host. And something else that's kind of unique about these filamentous phages is they can be extruded from the host without having to lyse and kill the host. So they can just be extruded out, just like pili are extended. It's a very similar mechanism. So I think I'll just kind of step back in time and tell you how and why I started studying phages, and it wasn't on purpose. Um, I started off really interested in how like human lung polymers would affect Pseudomonas biofilm formation. And some experiments that I was doing was to add this matrix polymer called hyaluronan. It's abundant in all of your tissues, your skin and your airways, your eyeball. And so I was interested in how that polymer interacted with Pseudomonas to modulate virulence potential. And so one of the first experiments that I did as a postdoc yeah, in Bill Park's lab at UW was to just grow Pseudomonas with and without hyaluronan. And so when you grow them in these tissue culture dishes, yeah, Pseudomonas just kind of forms this, flat, ubiquitous, boring lawn. But then in the cultures where hyaluronan was present, there were all of these crazy structures. And, this was like my first month as a postdoc in the lab. And I thought these things were like some sort of contamination that I brought into the lab. I thought they were like nematodes or something in my bacteria. And I was bringing this thing into the lab. But I repeated it, and I was really careful and was using sterile reagents, and these things just kept coming back, and after staring at them for a little while, it became pretty clear, that these things aren't alive, right? They never move really, and they're also crystal clear. You can see here that this one acts kind of like a lens. So eventually I did the experiment where I just scraped this entire mess up and I spun out all the bacteria and I filtered it through a 0.2 micron filter, which is smaller than an individual bacterial cell. And what came out the other side were these massive structures. The scale bar here is 200 microns. So these things are 1,000 times bigger than the pore size in the filter that I just put them through. So at this point, I was pretty much hooked, and my original project, died, and I started studying the contamination of my project. And some of the first experiments we did were some proteomics, and the proteomics came back, and it was very clear that these things were made up of these filamentous phages because the most abundant protein in there was the coat protein to this phage. And these structures ended up being, they're essentially droplets of these phages that spontaneously align. And so these things spontaneously align due to entropic interactions with the polymers. So when they align like this, a couple of things happen. The major thing is that they actually compact into the smallest volume that they can. And so that gives the polymers more room to wiggle around in. And so the increase in entropy on the polymers is what's driving this process to be spontaneous. They also align together because that increases their entropy so that they can move like this with each other. And so instead of, banging into each other like this, if they all align in the same direction, it increases their movement. And so that also increases entropy of the system. So this is a completely spontaneous process. And here it is in a movie. This is 3 minutes sped up a bit, but these things, they just form, super quick. Trippy, yeah. It's very cool. So this is how I became a phage researcher, also slash soft matter physicist, but not really. We started studying these things, and we started off with hyaluronan, but then we moved to other polymers, and we found the similar structures found with a bunch of different biological polymers. So we tried DNA and mucins and other glycosaminoglycans. And the picture here is we tag the virus filamentous phage with a fluorescent molecule. It's Alexa 48, and we mixed it with human serum, and these big clot-like structures formed. So what are these structures? I told you that they're aligned and they're like little droplets of phage or aggregates of phage. But it turns out that if you get any filamentous molecule to align with other filamentous molecules, you generate a liquid crystal. And so that's just halfway between a solid and a liquid, right? It's a crystalline structure on two axes, but it's free to move in the third axis. And so that's the definition of a liquid crystal. And if you have your phone and some polarizing sunglasses with you, you can take your polarizing sunglasses and turn them in the right direction, and it'll block the light from your phone because it's shining polarized light at you. So, and your phone has an LCD screen. It's a liquid crystal display. So liquid crystals have these polarizing properties with light, and that's what you're seeing here with this false colored image. So the brighter the color, the more organized, the more crystalline the area is. So we took this special microscope that we had custom built for us, and we looked at these phages, and we also looked at colonies of Pseudomonas. And the top one here we cured of its phage infection, and the bottom one still has the phage. And when we looked at it with this polarizing microscope, you can see that the bottom one just has all of these super bright areas indicating that it's got this liquid crystalline structure, and it's the biofilm matrix, all of the slime and stuff that it secretes that's being organized into a liquid crystal by the phage. This is what it looks like without false color. So there's a few cool things about this image. So how I took this was I put a Petri dish on a piece of glass and put that between two cross polarizing lenses. And then I shined bright light, white light through them. And so outside of the bacteria, it's black because no light gets through the cross polarizers. But in the bacteria where you see it's super bright, that's where the liquid crystal biofilm is reorienting the polarization of the light so it can make it through both filters. And then if you were to spin the bacteria, it would actually look like it sparkles because there's different areas that are organized in different angles. And then you also notice these bigger colonies. And those colonies are interesting because they actually became phage resistant. And so they're no longer able to be infected by the phage. And so they kind of lose their liquid crystalline properties. So I mentioned these phages are negatively charged, and there's a lot of them in these biofilms. Up to, in the lab, we can get them to produce up to 10 to the 11th virions per mil. So that's just an incredible amount of phage that can be produced by Pseudomonas. And they have a negative charge. So we hypothesize, well, maybe they can bind cationic antimicrobials. So we tested that by forming these liquid crystal droplets with the phage, and we mixed in this fluorescently tagged antibiotic called tobramycin, and it's a cationic drug, so it has a positive charge, and you can see that these liquid crystalline droplets, they soak up the antibiotic from the bulk solution, and they kind of sequester it. Then when you do kill curves with Pseudomonas, so what you're looking at here is just colony forming units of bacteria. And then on the other axis is increasing concentration of tobramycin, the drug. You can see that the bacteria that are living in a liquid crystalline matrix are about a log or about 10 times more, have a higher survival rate than bacteria that don't have the liquid crystalline matrix. So the phages seem to be protecting Pseudomonas from this type of drug. So this is really exciting in the lab, but we really wanted to know if these things were clinically relevant. So here is where I started talking with a guy named Paul Boyke. He is a infectious disease clinician, and we met up in Washington. He's currently at Stanford. We've been working together for a long time, but he has access to some clinical samples. And so he got a bunch of sputum, and we developed this qPCR assay to measure phage copy numbers in sputum. And so what you're looking at here are patient populations that have a confirmed clinical Pseudomonas infection or patients that are not clinically infected with Pseudomonas. And we can detect around 10 to the 7th phage copies per mil of sputum in the patients that have Pseudomonas, but not in the patients that don't have Pseudomonas. And we see a similar thing in wounds. So here we're looking at phage counts per swab. So we're just taking like a big Q-tip and rubbing it on the wound and see how many phages we can get to stick to that. And again, it's about 10 to the 6 phages per swab, only in people with clinical Pseudomonas infection and not those that are not infected by Pseudomonas. So the phages are there and they're abundant at sites of infection in humans. So they might have some clinical relevance. Oh, there's Paul. So Paul has this wound model set up in his lab where he does punch biopsies on the backs of mice. And so we got this model up and running, and we infected the mice with Pseudomonas. And we did this with 2 different strains. One was the wild type PA01 strain that has the PF phage integrated into its chromosome. And we did it with an isogenic Pseudomonas strain where we deleted the prophage from the Pseudomonas genome. And this is what we saw. So this is the wound after 6 days. You can see it's red, inflamed. It's not healing. It's not looking good. Whereas the mice that are infected with the strain that was cured of its PF phage, it's fighting off the infection. It's starting to resolve. It's starting to heal. Hair is coming back. It's really stark phenotype. So we started to dig into this, because it was really interesting. And I'm essentially going to summarize Yolene Swear's entire PhD dissertation in a single slide. But this is all her work. We lent a hand, but she did the heavy lifting here. So we know that Pseudomonas makes a bunch of filamentous phage. And what Yolene discovered was that when these filamentous phages encounter a macrophage, they stimulate a type TLR3 signaling. And so what happens is these phages are internalized by endocytosis. They engage TLR3s, they signal through TRIF, and they induce an antiviral response. So the macrophage or the dendritic cell, we tested both types. Both of them, antiviral immune interferon, type 1 interferon antiviral immune responses are triggered by these filamentous phage and these immune cells. And essentially what that does is it's like a decoy, or it tricks the immune system into thinking it's fighting a virus, but it's really should be mounting an antibacterial immune response. So it's a maladaptive immune response that allows Pseudomonas to set up shop and initiate and establish an infection. So the next thing that we did was, well, we hypothesized that maybe we could, break this cycle by of targeting the phages somehow. And the way that we did that was we developed this model so that we could incorporate longitudinal studies into this. So we did that by introducing luminescent Pseudomonas so that we could do some live animal imaging over time without having to sack the animals. And then we established an infectious dose where half the animals got an established infection, and we infected with wild type bacteria here. And then we did it with the Delta strain, and it went down. And then we complemented by infecting with purified virions, and it went back up. So now we have a working system where we can test some therapeutic interventions. And the therapeutic intervention that we tested was vaccinating against the major coat protein of Pseudomonas. So we did that. We had this adjuvanted peptide conjugate vaccine that targets the coat protein of the filamentous phage. And then we did the wounding, and then we did the experiment over time. And in mock vaccinated animals, again, about half of them established an infection, whereas the ones that were vaccinated against the phage, we saw a 40% reduction in established infections. What we also saw was Paul was able to make a monoclonal antibody against this phage, and when treated topically with the bacteria, the monoclonal antibody, when compared to a non-specific IgG, was also able to rescue or prevent infection initiation in this wound model. So how can immunity against the bacteriophage that infects the bacteria protect an animal host from infection? And this is something that we're actively working on. But we have a few ideas. So we know that antibodies, they can facilitate opsonization. And so that's just, it promotes phagocytosis of the bacteria, bacterial uptake. So maybe that's one way. Another possibility is that they are just binding to the virions and they're somehow manipulating how these viruses interact with TLRs or other pattern recognition receptors on phagocytes. And we also know that these antibodies disrupt the PF life cycle. And so a combination of these things is likely at play here to explain how this vaccine might work. And we are optimizing this vaccine now. We are working with a company here in Montana called Inimmune, and we have these adjuvants that we're trying to make formulations with. And we are testing those in different challenge models, and we're interacting with Chandan Sen's lab. He actually was at Indiana. He recently moved to Pitt, but he's got a pig wound model up and running that we're working with him on. And hopefully these things can get to phase 1 trials. So fingers crossed, that's the long-term goal. So all that is an awesome example of, like, basic bench to bedside translational science, right? But my passion lies with the basic science. That's where I'm really interested, and what I think about at night, right? And so, laying awake at night, one of the things that I'm thinking about is, these PF phages, they infect Pseudomonas clinical isolates. But if you look at the genomes of environmental strains, like, they're also there as well. They're not enriched in clinical isolates. And Pseudomonas, like I said, is an opportunistic pathogen. You can imagine that most Pseudomonas are like living out in the dirt or in the water or something, and they might never or rarely encounter a vertebrate host. So is it really interactions with the vertebrate immune system that is driving these types of mutualistic interactions between this phage and its bacterial host? I don't know. This doesn't seem like it's happening frequently enough in nature to kind of drive that type of interaction. So, laying awake at night, what else could drive these type of mutualistic interactions between this bacterium and its prophage? And we started thinking about, well, what are natural predators of bacteria? And one of those is are roundworms. Things like C. elegans, which is a nematode. And so C. elegans, or just nematodes in general, they're essentially ubiquitous in the environment as well. They're found in all niches in the environment, thousands of different species. And what's interesting about these things is that vertebrate innate immune pathways share evolutionary roots with the immune pathways in nematodes. So they share kind of some old pathways. So we hypothesize, well, these phages, maybe it's interactions with these kind of natural bacterial predators that's driving these interactions. And so we tested this in a C. elegans model in the lab. And this is where work from my graduate student, Caleb Schwarzkopf, comes in. He graduated last year now, 2023. And so this is his work. So one of the experiments that Caleb did was he took Pseudomonas and he grew them on the Petri dishes that we use to maintain C. elegans. And he found that wild type and the Delta strain, they don't have any growth defects and they're not making any phage, as indicated by this plaque assay here. So he repeated the same experiment, except this time he added C. elegans to the dish. And this time when he looked at the bacteria, he found that fewer Delta PF4 bacteria were recovered compared to wild type. And then the wild type bacteria started making phage. So the predation by the C. elegans is somehow turning on the PF replication in this model system. So remember this image with the green phages mixed with serum and how it made this clot? We see something similar in the intestines of the C. elegans, the worms, when they are fed fluorescently labeled filamentous phage. So this might be one way that these phages are helping out the Pseudomonas, is just by gumming up their guts and preventing the worm from eating the bacterial population. And then we also did some kill curves. So here we're just looking at Pseudomonas with its phage, and after 5 days, everyone dies. The graph doesn't go all the way down to the bottom because we weren't sure. The way we do this is we count the dead worms every day. And so some of the days we couldn't find their dead bodies. And so we weren't sure if they died or if they escaped. So they were withdrawn from the study. But if we compare this to the bacteria that have the phage, the survival curve is extended by 3 days. Everyone still dies, so Pseudomonas is still a pathogen. It's got a bunch of virulence factors beyond phage, but it just takes longer, significantly longer. So this is interesting. So to get a better idea of what was going on, we performed quantitative proteomics with worms collected at day 2. And Diane Brooks did these experiments in my lab. So we exposed them to bacteria for 48 hours. We ground their bones into dust, and we made sure that the proteins looked good. We ran them out on the gel, and then we did quantitative proteomics with this National Resource for Quantitative Proteomics. It's down at the University of Arkansas. They're an NIGMS center. And essentially what we do is we take our 8 samples. We have 4 replicates, 2 conditions. We digest the proteins and the peptides, and we label them with these mass tags. And you'll notice the colorful tags go up by a certain number, and the gray tags go down. So if you add these together, they all equal 305. So it's the same mass. And so then we can pool everything into the same tube, separate the peptides on LCMS, and then the things that come out in the single peak will be the same protein with the mass tag. And then when those proteins are fragmented to get an ID, what you get is you'll get a fingerprint, so you can identify the amino acids the peptide was made up of. And you'll also get these mass tags, which is now broken apart from the gray tag, and they're separated. And so we can actually determine the abundance of each peptide in each sample using this approach. So we got the data back and it looked beautiful. Here's just some gut check data. The peptide abundance was comparable across all the examples. The 2 groups separated when we looked at them on PCA. And then when we started digging into the data, we found all kinds of cool stuff. So Dominic Faith is another graduate student in my lab, and he spearheaded the analysis pipeline. He essentially just adapted our RNA-seq pipeline for proteomics. And one thing that we saw was the presence of collagen and extracellular matrix proteins. And these were significantly enriched when we did a gene ontology analysis. And this was interesting because the C. elegans cuticle, or its skin, is made up mostly of collagen. So we thought that, well, maybe the Pseudomonas that has the phage is, somehow disrupting cuticle integrity, which would be another way that would, kill the worm. So we collaborated with Katja Voronina and Mary Ellenbecker here at the University of Montana. They're a C. elegans genetics lab. And we did this really cool and simple assay. I love it because it's simple and it's got some cool images. But all we did was we added a nucleic acid stain to the worms and then treated them with bacteria or not. Or excuse me, we treated them with the wild type bacteria or the Delta strain. And then we just looked and saw if the nucleic acid stain was staining the worm's genome. And what we saw was, yeah, it was. So when this worm was exposed to Pseudomonas that has the phage, and you can see that the nucleic acid stain is penetrating the worm's cuticle and it's staining its nuclei. Whereas this worm, when it was exposed to the Delta strain, the only fluorescence we're seeing is from the worm, drinking up the dye from the liquid media that it was in. So, this is a confirmation that cuticle integrity is compromised, and it's dependent somehow on the phage. Another thing that we saw was the enrichment of proteins related to respiration and electron transport. And this really caught our eye because it relates to this other observation that we made early on with this Delta strain, and that is the production of this green pigment called pyocyanin. So we're able to extract this pyocyanin. We got a nice extraction assay for it and quantify it, and you can see that the Delta strain produces a lot more of it. And why this is interesting and why it relates back to those respiration proteins is that pyocyanin is a redox active molecule. So shown here in this test tube is this culture of Pseudomonas that's making a bunch of pyocyanin. And if you shake it, it's all green. But if you let it sit, the green goes away in the bottom and it remains on the top. And so this is an indicator of how far oxygen can penetrate the culture before it's all consumed. And so what the pyocyanin is doing here is it's acting as kind of like a breathing tube for the bacteria at the bottom. You know, they don't have any oxygen to breathe down here. And so what they're doing is they're dumping off terminal electrons onto pyocyanin to maintain redox balance. And then that pyocyanin diffuses away and dumps its electron off on a terminal acceptor that's somewhere else. And this is how bacteria can maintain redox homeostasis and form these big mushroom biofilm things and still be able to breathe on the inside or deep within where there's not much oxygen. So, you know, that's this molecule's purpose for physiology and everything, but this molecule also moonlights as a virulence factor. Because it's a greasy molecule, it can just diffuse right into immune cells. And once it gets into immune cells, it starts, it starts doing their redox cycle, but an immune cell that's not ready to handle that, it produces a lot of reactive oxygen species that ends up in cell death. So laying awake again at night, thinking the strain of Pseudomonas that has the phage doesn't make much pyocyanin, but the Delta strain does. It makes more of this virulence factor. But the one on the left that makes less virulence factor is more virulent than the one on the right that makes more virulence factor. What's going on? And we thought long and hard about this for a long time. Caleb thought about this for a long time too. Allison's in the audience. You might remember this project. So some clues came again from the proteomics data. We went back to the proteomics data, and another group of proteins that was enriched were these cytochrome P450 proteins. And these proteins are interesting because they are regulated by this receptor in animal cells called the aromatic hydrocarbon receptor. And so this receptor binds a bunch of different ligands. The thing that relates these ligands to each other is that they're aromatic, they're greasy. And so AHR, when it binds something greasy, it'll go and it'll turn on transcription of genes like cytochrome P450s, which have this heme core. And the purpose of these enzymes, you know, they do a lot of stuff, but one of the things that they do is help degrade some of these aromatic compounds that the cell might encounter. There's another paper out there, or a couple papers actually, by Mora Alves, and I really love this paper because it links pigment sensing, bacterial pigment sensing to host monitor of bacterial burden. So here is pyocyanin, and it's produced when you start getting a lot of bacteria around. And essentially what this paper found was that pigments like pyocyanin engage the AHR receptor and regulate host defenses to protect against Pseudomonas infection. And this paper found it in human cell lines, fish, and mice. So we started thinking, well, maybe the same thing might be going on in worms. So plugging what we know now into this model, maybe it makes a little more sense. So the strain that makes a bunch of this pigment activates AHR, host defenses are on, infection resolves. Whereas maybe if the phage is somehow tamping down production of this pigment, it can fly under the radar a bit longer, establish itself, and now it can produce, or it can cause disease in our model systems. So we predicted that if we disable the AHR signaling in the worm, that it might increase susceptibility to the phage or the bacteria that don't have the phage. So if we knock that out, maybe now this one will become more virulent. Where are we at? There it goes. So we did some more kill curves, and we collaborated with the Voronino lab again, and we got this AHR mutant here. And so we infected wild-type C. elegans with the AHR mutant with wild-type Pseudomonas that has the phage, no difference. We infected wild-type C. elegans with the bacteria that don't have the phage, and again, we see reduced killing. But this time, when we infected the AHR mutant with the Delta PF strain, we see that virulence is partially restored, indicating that the AHR pathway is somehow regulating host defenses in response to Pseudomonas pyocyanin. So here's all this kind of wrapped up in a nice graphical image. We got the PF prophage that's inhibiting pyocyanin production. And then over here, if it's gone, the bacteria just makes a bunch of pyocyanin, which activates the worm's defenses and reduces killing of the worm. So now we're going to get back into a little bit more of the molecular stuff. We started asking, how might the phage, inhibit pyocyanin production in Pseudomonas? So we went back to kind of just, looking at the plates and the cultures and stuff for the green phenotype, right? So we know if we delete the PF prophage, the bacteria turn green. The opposite is true. If we infect the bacteria with purified virus, the green goes away, and that's quantified here. So we did some RNA-seq on these cultures infected with PF virions, and the most highly upregulated set of genes, not surprisingly, belong to the phage. But if we take those out and look at everything else, one thing that really caught our eye was that pyocyanin biosynthesis and quorum sensing genes were all downregulated in this culture here that's infected with phage. So quorum sensing, when I teach about quorum sensing in my class, I relate it to body odor. So like if you're in a room by yourself, maybe with your roommate or something, you can stink a little bit. It's not going to change anything. But if you get into a room with a whole lot of people, it is crowded, it is hot, it is sweaty, people are smelling bad, it's going to change population dynamics, right? People are going to start behaving differently. And so bacteria do the same thing. So they start, producing all of these molecules. This one here is called PQS. It's the Pseudomonas quinolone signal. And it is made by the PQS operon, and it looks like that. And when this thing is present at high enough concentrations, it will bind to the PQS receptor, and that promotes, the receptor will bind to promoter regions in the chromosome. And this allows Pseudomonas populations to regulate gene expression at the population level. And that includes regulation of secretion of things like pyocyanin and other virulence factors. So for these next experiments, we collaborated with Ajay Dandekar at the University of Washington. He studies Pseudomonas socio-microbiology and CF pathogens like Pseudomonas. And Pseudomonas, our lab strain, it has 3 primary quorum sensing systems. It has LAS, REL, and PQS. And so Ajay has these fluorescent transcriptional reporters for each of them. And when we tested them all, we didn't see a difference in LAS and REL, but we did see a difference in PQS when we measured quorum sensing transcription in wild type versus the Delta strain. So when we delete the prophage, this PQS transcription goes up. So that suggests that there's a protein that's in the phage that is somehow keeping it down. So the nice thing about working with this phage is it's tiny, right? It's only 12 kb. It's got a handful of genes. And so we can do genome-wide screens, kind of brute force. So Caleb, here he is again, went through the entire genome with the PF phage integrated into the Pseudomonas chromosome. And what he did was he expressed each gene from a plasmid in the strain that's missing the PF prophage. And then he measured pyocyanin production. And he found that there was one gene that significantly suppressed pyocyanin production compared to the Delta strain without expressing a phage protein. So this hit came back as this protein called PFSE, which was really surprising to us because it was a protein that we published on a couple years now. So this was Amelia Schmidt's work. She's another grad student in the lab, and she discovered this protein and named it, and she named it PFSE for PF super infection exclusion. And what she found was that this protein was essential for inhibiting type 4 pili expression to prevent Pseudomonas from becoming reinfected by the same phage. And she did a lot of cool work, but essentially PFSE is an intermembrane protein, and it will bind to the PIL-C protein in the type 4 pili apparatus, and it'll essentially just gum it up, and it prevents the pili from being extended, and so that prevents the phages from being able to use them to get back into the cell. So, this was kind of surprising to us because we weren't expecting an intermembrane protein to be able to inhibit pyocyanin production by Pseudomonas aeruginosa, and that's because most of the biosynthetic proteins that make pyocyanin are cytoplasmic. They're soluble proteins. So to try and answer this question, we teamed up with Karen Maxwell and Vero Taylor up at the University of Toronto, and they had this awesome bacterial 2 hybrid system set up to look at protein-protein interactions. And we've already worked with them with Amelia's project and to discover that this phage protein binds to PIL-C, which you can see here. So in essentially this assay, how it works is if two proteins stick together, the bacteria make a pigment and they turn dark. So we were able to use the PIL-C as a positive control. This is the empty vector negative control. And Karen and Vero just happened to have a whole bunch of quorum sensing genes already cloned and ready to go. And so we just started trying them all, and we got lucky, which is super important in science, right? We got lucky and we found one in PQSA. It was another protein that caused pigment production, indicating an interaction between these two proteins. And we quantified that here. So what's PQSA? PQSA is the first enzyme that synthesizes the PQS quorum sensing signal. And so it turns anthranilic acid, this aromatic compound, after a few steps, many steps, it turns it into PQS. This structure has been studied for a long time. It's got a nice protein structure. It's bound to different ligands and all this other stuff. So we hypothesized, well, maybe, anthranilic acid is kind of hydrophobic. It's got that phenol ring. Maybe the hydrophobic intermembrane protein is sticking to this PQSA protein in inhibiting it from producing PQS signal. So we're working with Levi McClelland here at the University of Montana. We got some crystals of the protein complex made, and we're waiting for beam time. And hopefully someday soon we'll have a solved structure. But until then, we have AlphaFold to play with. And so we plugged these sequences into AlphaFold and asked it to fold them up for us and predict how these two proteins might fit together. And we use the solved PQSA structure as kind of a starting point. So here's the solved structure, and this is what AlphaFold did. Instead of just laying the protein on there, it actually folded the phage protein in half and then stuck that into the catalytic domain. And you'll notice over here that the aromatic residues, they reach deep into the catalytic pocket. So if you take PQS away and look at just the structure of the phage protein, there's a lot of interesting stuff going on here. One is this central glycine in pink. It's conserved across all PFSE proteins, and glycine, its side chain is just a proton, right? So it's the smallest amino acid, and so that's where a lot of proteins fold in half. And so that's what's happening here with ours. Something else to notice is that all the aromatic amino acids in yellow are on one side of the protein in this conformation. And then a third thing to notice is that at either end of this protein is an acidic or a basic residue, and those might serve to stabilize this conformation to keep it folded in half like that. So hopefully our structural studies will confirm this, but it's making a lot of intuitive sense for now. So let's take what we've learned, and we'll plug it back in our model. So the prophage makes this protein, PFSE, and we know that the protein inhibits type 4 pili, and we also know that it inhibits PQS. And this is really interesting because Karen Maxwell's lab also found a phage protein. This is a different phage that infects Pseudomonas. It encodes this protein called AQS1 that inhibits type 4 pili by binding to a different protein in the type 4 pili complex. And it also inhibits a different quorum sensing pathway. Same protein doing the same thing. So this is a nice example of convergent evolution, we think. This might be a common viral strategy. It might be somehow important to inhibit quorum sensing and prevent other phages from coming in. And we think that this might be related to our observation that inhibition of PQS quorum sensing actually increases Pseudomonas's susceptibility to phage infection. So we're starting to think of quorum sensing as more and more of kind of like an innate immune pathway in bacteria. We think quorum sensing regulates a lot of anti-phage defense mechanisms. And if you disable it, Pseudomonas becomes more susceptible to phage infection. So essentially, by having a protein that inhibits quorum sensing, you're making your host more sensitive, more susceptible to competitors. And to combat that, you also take away one of the major cell surface receptors that phages use to get into the host cell. So to wrap this all up, I always like pointing out that this phage has this super simple morphology, this simple, tiny little genome, but it has this incredibly complex biology. It does all of these interesting things. So I just love pointing that out to my students and stuff. You never know what you're going to find. So I've thanked a lot of folks along the way, and there's a lot of folks in my lab too who are working on other phage stuff. We got E. coli projects and Lyme disease projects going on as well. So if you're interested in that, I'd love to chat about that as well. So yeah, with that, thanks for your attention.
Okay, thanks, Pat. So